Welcome to our video lecture on topic B3.2, transport in plants. There's a separate video for transport in animals. This video is both core and some additional higher level content. SL friends, I will let you know when we get to the AHL slides so that you can ignore them should you choose. Our guiding questions: what adaptations facilitate transport of fluids in animals and plants? We're focusing on the plants. One of our last slides is going to be a lovely Venn diagram in which we will look at the differences and similarities between transport in animals and the plants that we're talking about today. Our objectives: we're going to explain transpiration. We did introduce this in topic B3.1, gas exchange in plants. We're going to add lots more details today. We're going to outline different methods for measuring the rates of transpiration in plants. We're going to talk about the distribution of tissues in dicotyledonous plant stems and roots. This is just one category of one kind of plant. It's a common kind of plant, so IB has chosen it to be our model plant. We're going to talk about some adaptations for xylem in transport. We're going to, as I just mentioned, look at that lovely Venn diagram, similarities and differences between transport in animals and in plants. We are, for my higher level friends, also going to add some more details regarding the propagation of xylem root pressure. We're also going to talk about translocation, which is what happens in phloem. And again, we introduced the idea of transpiration in our B3.1 gas exchange in plants video lecture. So feel free to go back and think about that if you want to. That B3.1 gas exchange video lecture talked mostly about what's happening in the leaves. Here, where we have a higher concentration of water vapor in the leaves, that lovely spongy mesophyll, and that allows for the diffusion of that water vapor out of the leaf tissues through the stomata that can be opened and closed by the guard cells. Again, if you want more details on that, go back to topic B3.1, gas exchange in plants. We're going to focus now a lot on how do we get that water to move up the tree or the plant against the force of gravity. It is done in some lovely tissue known as xylem. Xylem's kind of cool in that it's dead. So those cells, once we have that mature xylem, those cells are not living. They don't have cytoplasm, they don't have cell membranes, they don't have mitochondria or nuclei. All that really is left are some super cool cell walls. Those cell walls are made of cellulose. Cellulose is a little bit polar, my friends, and that means that we can propagate some adhesion between our water molecules and the cellulose cell walls of the xylem. That adhesion, that force of attraction between the polar water and the polar cellulose, allows for the water to get kind of sticky. And then also, remember that water, because water is polar, we have some cohesion, that force of attraction between the water molecules. This leads to lots of beautiful capillary action. Capillary action is that movement of water up a thin tube. In this case, our thin tube is the xylem. Because the water is attracted a little bit to the xylem and then also to itself. And because we have that water leaving here, when this water molecule leaves the leaf, it pulls another water molecule up, which pulls another water molecule up. And because we've got stickiness here and stickiness here, that water can move up the xylem in a constant stream. And we call that the transpiration stream. It's this lovely continuous chain of water molecules that can move from the roots up the xylem and then out of the leaves. Not just water is going to be moving through the xylem. We're also going to dissolve in the water some minerals. We're going to call it the xylem sap. The sap in the xylem is mostly water, but those minerals, maybe some potassium, some chloride, some magnesium, whatever other minerals might be in the water, in the soil that we can dissolve in the water that the plant will need for enzymes and whatnot. We can measure the rate of transpiration with some super cool devices known as potometers. Potometers, lots of different variations of these. We're going to build some in lab, so getting excited about that. So this potometer here, we have a pipette. It's calibrated so that I can measure the level of the water. We're going to have some flexible tubing that's filled with water. where we have our plant here that we have stuck into the tube with the water. What's going to happen is as the water exits the leaves due to transpiration, it's going to pull some more water up through the tube, which is going to pull some water out of that pipette. And we can measure the falling of the water level here. The water is going to go down because it's going that way. So as that water level decreases here in the pipette, we can record, because we're looking for rate over time. Rate is all about how fast is that stuff going, how much volume of water is being lost through the plant over time. We can also have this much fancier one here. Again, we've got our plant cutting, we've got some water, we have this lovely scale next to our capillary tube. We're actually going to introduce an air bubble here, and then we can record the distance that the air bubble is moving, because it's moving with the water, as it's going that way toward the plant, being lost out of the stomata of the plant through transpiration. Again, we're going to do this over time because we're looking for rate. This one, a little bit simpler. We're going to put the graduated cylinder with some water and the plant on a scale. We're going to measure the initial mass of all of this. We're adding to the top of the water a little layer of oil. That's going to prevent the evaporation of the water out of the graduated cylinder, so that the only water loss, the only mass loss, is going to be the water that is exiting through the leaves here. And again, we're going to measure the change in mass over time, because rate is all about some change over time. We're going to put function away for a little bit and focus on structure. Instead, we're going to focus on the structure of tissues in stems of plants. IB has chosen to focus on plants known as dicotyledonous plants. We can say dicot for short. There are lots of different kinds of plants, and so not every stem is going to look like this, right? But this is a really nice common kind of plant, and so we're going to focus on these dicots. Not all plants look like this, but some do. So these are dicots, dicotyledonous plants, and this is a cross section through a stem. We're going to look at roots in a couple slides. So here in the stem, the outer layer is the epidermis. It's kind of like the skin of the plant. Its job is protection. Its job is to prevent water loss. We have in between the epidermis and then the inside, which is the pith. The job of the pith is just support and storage. We have some cortex. The cortex is like this filler stuff between the epidermis and the pith. The cortex, again, is just support and maybe a little bit of photosynthesis if it is a green stem. The coolest stuff that we're going to look at here are these chunks known as vascular bundles. Just like in us animals, we have our vascular tissue, veins and arteries and capillaries. Plants also have some vascular tissue. They are going to bundle their vascular tissue into these super cool vascular bundles. Within each vascular bundle, there is some xylem and there is some phloem. Here we have a zoom in of just one of those vascular bundles. Here's the xylem. Notice that the xylem is quite a bit larger. So the xylem cells, and again, we use cell loosely because xylem is actually dead. Those xylem columns are quite a bit more open. The phloem is smaller. And on the other side of the phloem, we have some supporting fibers. I'm going to add a tissue that's not labeled in these diagrams. Between the xylem and the phloem, there is some cambium. Cambium is a kind of meristem. Meristem is the stem cells of plants. And so we have some stem cell tissue in here between the xylem and the phloem. And of course, the job of this cambium is to produce more xylem and phloem tissues. The job of the xylem is to transport the water and the minerals that we were just talking about. The job of the phloem is to transport organic materials. We're going to focus mostly on sugars, carbohydrates that are formed during photosynthesis. Notice that in our vascular bundles, the phloem is closer to the outside of the stem. The xylem is closer to the inside of the stem. phloem toward the outside, xylem toward the inside. Xylem is bigger. Those cells are just more open. And again, cells, they're dead, the columns. And then the phloem is smaller, more toward the outside. Also, when we collect maple syrup, what happens is we put our little tubes to the phloem, and then all of that sap, the sugar water that the plant is trying to store for itself, we humans go and steal. Good? All right. We're going to throw in here a quick little stem cell review. Remember that totipotent, these are cells that can become totally anything at all. Pluripotent stem cells can become so many things. Multi can become a couple things. Unipotent stem cells can differentiate into only one type of cell. What's pretty cool, that cambium, that meristem stem cell tissue in plants that's between the xylem and the phloem in the vascular bundles, it is pluripotent. Cambium is pluripotent. It can differentiate into many types of cells. It's mostly going to become xylem and phloem. Remember from our B3.1 gas exchange in plants lecture that plan diagrams are super non-detailed sketches showing just the distribution of tissues. We don't need to draw lots of individual cells. We're just going to show where those tissues would be. We need to be able to draw plan diagrams of dicot stems. We're going to start with, well, I'm going to start. You can draw however you like. I'm going to start with the epidermis. Woo, that's not a great circle. And then just inside the epidermis, I'm going to draw the cortex. We've got some cortex here. So again, epidermis, that outermost layer, and then just inside we've got our cortex. We need to add some vascular bundles. We're going to add some vascular bundles. They're in a ring just inside the cortex. And then inside our vascular bundle, we have some phloem more toward the outside of the stem and xylem more toward the inside of the stem. And then on the very inside of the stem, we've got that pith for storage. From stems to roots. The roots have some things in common with the stem structure. There is that layer of epidermis. Its job is protection, preventing water loss, just like in the stem. Under the epidermis, we have lots and lots of cortex. Cortex, just like in the stem, is here for support and storage. What's not going to happen in the cortex of the root, though, is photosynthesis. So sometimes stems can carry out some photosynthesis in their cortex cells. There is no need for photosynthesis to happen here because our roots are underground where there is no sun. It's really hard to do photosynthesis without that sunshine. So cortex of the root, not photosynthesis, but yes, support and storage. At the very center of the root, we have this one lovely vascular bundle. We're going to zoom in on that vascular bundle here. The larger, more open cells, structures, because they're not living cells anymore, is the xylem. So xylem here in the middle, kind of forming this X-shaped formation. And then the phloem is in those divots around the xylem. So we've got xylem, we've got phloem. Xylem still more toward the middle of the root, just like we had xylem more toward the middle of the stem. We do need to be able to draw a plan diagram of roots, just like we did of our stems. And so again, we're going to draw just a super non-detailed, no cells needed sketch to show that roots have epidermis and cortex. And then we've got that lovely vascular bundle in the middle, kind of an X shape of xylem, and then phloem between the legs of the xylem. And again, that's one very beautiful vascular bundle in the middle of our root. And we're going to zoom in even more on those vascular bundles and look at the xylem itself. So those lovely big open cells that are more toward the center of the stem, more toward the center of the root, the job of the xylem, again, is to transport water, lots and lots of water and minerals. What's pretty cool about that xylem, I've mentioned, it's not really cells anymore because they're dead, but they started off as cells, so they had cell walls and cell membranes and some cytoplasm and nuclei, all kinds of super cool stuff. But then as the xylem matured, those cells actually died. So the cytoplasm went away, the cell membranes went away, the nuclei, the mitochondria, everything went away. What we had left were cellulose cell walls. The super cool plants, not us humans. So the plants take away the cell walls between the cells, and that results in this lovely continuous column for which water to be transported. Also added to these dead cellulose columns is these rings of material called lignin. So lignin is super cool. It's what these rings are. These rings of lignin are going to add some strength to the xylem. The xylem is going to be under some lower pressure, so pressure lower than atmospheric pressure. We don't want the xylem to collapse because then the water can't get transported up the plant. And so this lignin is just a reinforcing layer. It's going to help keep that xylem open. It's also going to help support the stem as it grows up against the force of gravity. What's interesting is that the lignin is hydrophobic, and so the water is not going to want to stick to it. And so it's not a continuous layer of lignin. Instead, it's those rings. So we have lovely hydrophilic cellulose, lots of it, and then there's just little rings of lignin. And again, that lignin is hydrophobic, nonpolar, whereas our cellulose that makes up the majority of the wall of the xylem is going to be hydrophilic. Trees that have crazy big tree trunks and grow super, super tall and need lots and lots of support to stay super tall are going to have more lignin. But they're going to add some pits in the cellulose and the lignin, just to allow for that water movement to continue to happen, because too much lignin is going to reduce that ability of water to flow. The next two slides, this one and the next one, are additional higher level content. So my HL friends, you must continue paying attention. You will be assessed against this knowledge. SL friends, you don't have to listen, but it's kind of cool. So it's not going to hurt you. SL friends, though, do not stop watching the video. Don't call it done. Instead, skip ahead a couple minutes so that you get that Venn diagram that is looking at the similarities and differences in transport between animals and plants. HL friends, let's talk about how do we get an increased pressure in the roots xylem. We've been talking a lot about transpiration, that movement of water up the plant against the force of gravity. We talked about the structure of the xylem, how it is adapted to carry out that function of water movement. Let's talk about the roots. How do we even get the water in the roots so that we can transport it up the plant and to the leaves? So lots of adaptations in the roots to allow for this to happen. So many branches, lots of branches means more surface area. More surface area means more absorption. Roots also have some super cool little projections called root hairs. Root hairs, more surface area, more absorption. Just like our alveoli, just like our capillaries, all these other adaptations that living things have to increase surface area for more absorption. This is what the root hairs and the root branches are doing as well. Pretty cool thing happens. So the root is actually going to actively transport some hydrogen ions out of its tissues. Those hydrogen ions are going to displace minerals that are stuck to soil particles. That makes those mineral ions, mostly they're going to be ions. There might be a few molecules, but mostly ions. Those ions are going to then be dissolved in water, and then some of those minerals are going to be actively transported into the plant. Some of them will diffuse in due to some concentration gradient differences. The magical thing, though, about getting the minerals into the plant, you guessed it, the plant root is now hypertonic. And because the plant root is hypertonic, water is going to flow via osmosis from the environment into the root. So a little bit of active transport of those hydrogen ions, a little bit of active transport of some minerals is going to lead to the passive osmosis of water into the root. What's kind of cool is that that can happen even without water evaporating from the stomata of the leaves. So even if we close down the stomata, even if there isn't any water evaporating, leading to a transpiration pull, we still have a push because that hypertonic environment inside the root is going to lead to the osmosis of water into the root. All about that pressure. That pressure again can help to push the water up. Transpiration will help to pull the water up. And we have talked so many words about xylem. It is finally the turn for some phloem details. When we talk about phloem here, the job of phloem is translocation. Translocation is the movement of organic molecules around the plant. The organic materials we're going to talk about the most are the sugars that are formed during photosynthesis. They're going to go from a source to a sink. That is the direction of transportation in translocation in phloem. Remember that in transpiration, we always went up the plant. It was always from the roots, through the stem, out the leaves. Translocation in the phloem, we're going to go from the source to the sink. The source could be a leaf cell where we are making lots of sugar during photosynthesis. And perhaps the sink is a root cell. Maybe this is a potato and we're going to store lots of starch in the potato. So our sugars are going to move from the source, the leaf cell, to the sink, the potato tuber root cell. But know that that isn't always the case. The source isn't always a leaf. The source could be, are you ready for this? The source could be the potato. Because what if I plant a potato that used to be the sink, but now it's the source, and we're using the organic materials that are stored in the potato to grow a new potato plant. Now this is going to be the source. The potato is the source because we're moving the material out of the potato and we're sending it to the growing leaves. So we totally flipped the source and the sink. So in our transpiration xylem tissues, we always move up, up, up. Translocation, we could move down, but we could also move up. So translocation is going to move always from source to sink. But how? Let's talk about these details. So here, this example, we've got our source cell. In this case, it is the leaf. It's making lots of sucrose. That sucrose is going to get transported into some companion cells. The companion cells are then going to transport the sucrose into the sieve tube elements of the phloem. So phloem consists of two kinds of cells, sieve tube elements and companion cells. These sieve tube elements are pretty cool. They are almost dead, but not all the way dead like xylem, but almost. So they do have a tiny little bit of cytoplasm left. They do have a plasma membrane. This micrograph is showing sieve tube element cells. You can see we've got cellulose cell wall here. There's a tiny, tiny little bit of cellular material, but it is mostly not there. It's mostly empty space. These companion cells, the jobs of the companion cells, CC for companion cell, is to load that material into the sieve tube elements, but it's also to keep those sieve tube element cells alive because they're almost dead. They don't have any mitochondria, they don't have nuclei left anymore, but the companion cells can support the sieve tube cells. These sieve tube elements, they do have, unlike the xylem, they've got some more cell walls left between them, but they've got super amazing giant plasmodesmata, which I feel sounds kind of evil, right? Like, oh, it's Dr. Plasmodesmata here to take over the world. Actually, they're not evil at all. Plasmodesmata are simply these pores in the cell walls that allow for the movement of fluids between cells. So we've got plasmodesmata between the sieve tube elements. We also have plasmodesmata between the companion cells and the sieve tube elements, so that we can move all of that super cool organic material into the sieve elements, and then through the sieve tube elements to the sinks, whether that be an apple or a potato. What else did I want to say about this? Ooh, yeah, no, this is super cool. So once we get all that sugar into the sieve tube elements, we now have, are you ready for it? Another hypertonic environment being propagated. And you know what happens with hypertonic environments? Well, the water is going to move there via osmosis. You know where the water comes from? The xylem. You know how in all of our vascular bundles we have our xylem sitting right next to our phloem? Well, one of the benefits of this is that we built up our hypertonic environment because we've moved all this sugar into the sieve tube elements. Water from the xylem can flow via osmosis there as well. So now I've got lots of sugar and I've got lots of water. There's a lot of stuff right there. That lot of stuff is going to turn into a high pressure. We call this particular pressure hydrostatic pressure because it's lots of water. So that's a C, not an S. So that increase in hydrostatic pressure is going to push stuff away from this particular spot in the plant. We have a lower pressure here where the sink is because we're pulling the sugar out. Because we're pulling the sugar out, it's not hypertonic anymore, which means the water is going to get moved back to the xylem. So I have low pressure here at the sink and high pressure here at the source. And so all of this sap is going to move away from the source toward the sink due to those differences in hydrostatic pressures. Here's a little zoom in on a companion cell and its sieve tube element. Notice all the mitochondria, notice that lovely large central vacuole, notice lots of cytoplasm. All the things that a cell needs to stay alive are in the companion cell. Notice that sieve tube element. Oh my gosh, he's got his cell wall and that tiny little bit of cytoplasm in some cell membrane, and that's pretty much it. And so those companion cells are definitely going to help to keep those sieve tube elements alive. Notice here that arrow is pointing to a beautiful plasmodesmata, those pores in cell walls that allow for fluid to move pretty easily between some plant cells. Phloem is considered an atypical cell. These sieve tube elements are atypical in that they don't have nuclei and they have very little cytoplasm and they're almost not alive, but they are because they're friends, the companion cells. We talked about atypical cells a really long time ago. Let's talk about those other examples of atypical cells. So phloem are atypical cells. Other examples, our erythrocytes. Remember, that's our fancy word for red blood cell. Erythrocytes are also anucleate, don't have a nucleus, just like our sieve tube elements. Muscle cells are also considered atypical, those muscle fiber cells, because they are multinucleate and very, very large, as are our aseptate hyphae in fungus. These guys are atypical cells. They are cells, but just barely. They are atypical because they are multinucleate and also really, really, really big. And as promised, we're going to wrap up with our lovely Venn diagram comparing and contrasting transport in animals and in plants. There's actually a lot in common between us animals and plants. We both have a water-based fluid that we use to dissolve the things that we want to transport around our bodies. Of course, in us animals, that's going to be blood, and then the fluid in our lymphatic system. In plants, we're going to have xylem sap, and then we also have some phloem sap. In the xylem, we have, again, just water and minerals. In the phloem, we have water dissolving organic materials like sugars. Both plants and animals have some tube-like structures that are going to allow for that water-based fluid to get moved around. In us animals, we have our arteries, and we have veins, and we have capillaries. Plants have xylem and phloem. Both plants and animals use some pressure gradients to get the fluid to move around their bodies. In us humans, in us animals, we have hearts that pump really hard to get the high pressure in the arteries. There's a lower pressure in the veins, and so the blood will move through the arteries to the veins, taking that little pathway through the capillaries that allows for that nutrient exchange. In plants, we're going to have a high pressure in the root xylem and a lower pressure in the leaf xylem, and that is going to allow for that movement of water up against the force of gravity. In phloem, we have a high pressure at the source and then a low pressure at the sink. And again, that's going to allow for that material to move around the body of the plants. And both of these guys have some crazy amazing adaptations to increase surface area, lots of branches of our blood vessels, lots of separate tubes of xylem and of phloem. And then of course we've got the lovely root hairs in the roots to increase surface area for more absorption of water. The one big difference is that plants in the phloem, phloem, do have a two-way flow. So in us animals, it is all unidirectional movement away from the heart through the arteries to the capillaries, back to the heart through the veins, unidirectional flow of the blood. In plants, in the xylem, it's unidirectional. We're going from the roots up the plant to the leaves, but phloem can go whichever way it needs to go, always from the source to the sink. But that doesn't necessarily mean up or down or left or right. And on that note, my friends, we have achieved all of our objectives. We talked about transpiration, that movement of water into the root, up the stems, and out the leaves. We talked about how we can measure the rate of transpiration with potometers. We'll build some of those in lab. We talked about the distribution of tissues in dicotyledonous plant stems and roots. Be able to draw those plan diagrams. We talked about adaptations of the xylem for transport. Xylem is dead, lots of cellulose, lots of lignin, so that it can move that water up the plant against the force of gravity. And we built our Venn diagram. Our additional higher level content, we talked about how we can actively transport some hydrogen ions, some minerals, leading to a hypertonic environment in the root that leads to osmosis, and that increases the pressure of stuff in the root of plants. And then we talked about phloem. The function is translocation, and there are companion cells and sieve tube elements and super cool stuff. Great work today, my friends.